just finished up our unit looking at the organization of life, and we studied briefly that atoms are the smallest component here that make up obviously molecules, macromolecules, and then the smallest unit of life is a cell. And I really want to spend some time looking at cells because they're foundational to what we're studying in anatomy. If we don't have a proper understanding of the components of cells, so cell organelles, and how cells interact with each other and communicate across their cell membranes, we're not going to have a good understanding of what tissue is made up. What tissue is made up of. Remember that tissue is a group of similar cells working together. And then different types of tissue that work together form organs, and organs obviously form organ systems. So we need to start at the foundational point. And so let's just jump into our celebration here. Get it? No pun intended. Actually, pun intended. Anyway, human cells are animal cells, which are different depending on their function. But there are a lot of similarities, so think about it. Think of a liver cell, a heart cell, a neuron, which is, we're talking about brain cells, nerve cells. You can think of bone cells. All these cells are different, and we're going to look at how they're different in their structure, which makes them different in their function. But they have a lot of similarities in the organelles that they have. Every cell in our body, I do want you to recall, has the same DNA or genetic material. However, different parts of that DNA are coded in different cells, which we'll jump into. If you look at this image here, these are all animal cells. The far left one, the blue there, that's an amoeba. We don't have those in our body, but they are an animal cell. Obviously, we have a couple of these other ones in our bodies. The red blood cells, you can see at the bottom. You can see the nerve cell off to the right. And look at their structure. They're very different, and so structure plays a huge role in function. Looking at this animal cell, hopefully you'd be able to identify every part of the organelles here. If you look at the actual liver cell right here, you should be able to identify all those various organelles, and that's what I'm going to expect you to be able to do when we study the different cells we'll look at. So if you look at these two images side by side, you can see obviously the nucleus in the center of that liver cell. And you can obviously see the nucleolus at the center of the nucleus, like the purple there. You can see the red mitochondria in the liver. Liver has a lot of mitochondria. You can see the rough and smooth ER that's very close to the nucleus. It's the green there. The little blue dots, okay, those could be ribosomes. And the purple, we're looking at cytoplasm, and you can obviously see the border, the cell membrane. So hopefully you'd be confident in identifying those organelles in time. Let's recap on some of those organelles. So remember, the nucleus controls all cell activity, and it is the storehouse of all genetic information, obviously our DNA. So DNA is replicated here, and we also have transcription here, where DNA is transcribed into RNA. Remember, we eventually use that RNA. That RNA is eventually translated out in the cytoplasm at the ribosome to make protein. So the nucleus plays a very large role in the cell to actually store the DNA and also transfer genetic material. Analogy is kind of like a city hall, which we talked about in biology. Think of the control center, things that are getting done, that place where all that's happening. There's obviously the nucleus in the purple there in the center of that liver cell. The nucleolus, if you recall, is located in the nucleus, and this is where ribosomes are made, and they migrate out into the cytoplasm. Remember, ribosomes are where we make protein. So all protein in the body is actually produced in the ribosomes, which is very important. The cell membrane, you can see the divide here between the green and the blue there. Obviously, this protects the cell from the outside environment. It is selectively permeable. Remember, when you're selective, you're choosy. Permeable, you allow some things in, some things out, things can move across. So it controls what material moves in and out of the cell. It acts like a gatekeeper. Remember, the cell membrane is made up of phospholipids. We have hydrophobic heads, hydrophilic tails. Hydrophilic heads, water-loving heads, and hydrophobic tails, which allows the cell membrane to be selectively permeable. Some things obviously pass the cell membrane either through diffusion, osmosis. They're larger substances, they have to go through active transport like endocytosis or leave the cell through exocytosis. So we think of this as a gatekeeper or the city border of our city, kind of protecting the cell. Here's the cell membrane. Again, you can see those phospholipids here. And then you can obviously see there's membrane proteins here. There's a lot of protein-gated channels, which also goes back to active transport, allowing things in and out. Obviously, that uses energy. The cytoplasm, if you look here, it fills the space between the nucleus and the cell membrane. It is the fluid portion of the cell, and we call that fluid portion cytosol. It's mainly made up of water. This is where we see the site of most cell activity. So substances and important things are transported throughout the cell by means of the cytoplasm. They can go from one area of the cell to the other with the cytoplasm. This is kind of like the property or the city proper, where there's obviously a lot of movement on the property. Ribosomes are the place where RNA is translated to produce protein. So remember that RNA was originally transcribed from DNA in the nucleus, then it actually leaves the nucleus and is translated at the ribosome to actually produce protein out in the cytoplasm. Think of this as a lumber yard. You're taking something, you're taking wood or a tree, and you're actually making something different out of that. That's why we kind of have the lumber yard as our analogy. Endoplasmic reticulum, or as you know it, the ER. This is a network of folded membranes that transport materials, nutrients, and waste throughout the cell. There are two types, if you recall. We have rough ER and smooth ER. This is kind of like the highway road system, transporting things across the actual cell. Smooth ER, if you remember, lacks ribosomes, and it acts as a storage area for proteins exiting the cell. So that's really what's getting stored. There are proteins. The rough ER, remember, contains ribosomes. And remember, ribosomes make your protein. So the rough ER, I want you to think of like a rough road. You're traveling on a very rough road. Think of like a semi transporting things on a rough road. This is where proteins are transported in and out of the nucleus. Oops, I'm sorry, I'm going to go back there. So remember, smooth, think stores, rough, transportation of proteins. The Golgi apparatus is a condensed membrane enclosed space that processes, sorts, and delivers proteins created in the ribosome on the ER. So this is kind of the sorting area. Remember, this is like the post office or the UPS in our analogy. Here you can see the Golgi apparatus. Here's a blown up image of what it actually looks like in the cell. So you can see the Golgi apparatus come after the rough endoplasmic reticulum, or after that ER, and it really sorts and directs where that actually needs to go. The mitochondria, remember, you know it as the powerhouse of the cell, mighty mitochondria. This converts nutrients the cell intakes to usable form of energy. All things need energy, as we talked about, and your cells aren't excluded. It is folded and kind of like a kidney bean shaped when you look at it in all of our cells. Muscle cells have a lot of mitochondria. You need to convert nutrients to actually use as energy. Think about how much energy you're exerting when you flex. So analogy is like an energy plant or electrical plants. Here's a mitochondria. Don't worry about the folds and the cristae, the matrix. You don't need to worry about that. I just want you to remember its function. Lysosomes are formed in the Golgi apparatus. Remember where we had that processing, packaging, sorting, and delivering. And lysosomes actually break down food. Okay, they recycle things, they break down food, used and worn out cell parts, such as organelles. So just as your cells are always replicating because you need new cells because they're worn out, they're dying, you need new organelles. And so it actually breaks that down and restores those components to the cell when it breaks it down. So it returns those digested components to the cytoplasm, so then you can reuse those components. This is kind of like the recycling center of the cell. All right, we have microtubules, and we're almost done. This is a network of proteins that give the cell support, and they're found in the cytoplasm. It helps give the cell shape and mobility. And then it's kind of like the brick foundation of a city, or like of a home. Cilia, obviously this is not found on all cells here. We'll definitely look at cilia in the reproductive tract. We'll look at cilia when we actually study the respiratory system. And these are small beating hair-like extensions that are used to create a current to move things things away and out of the cell. So they're actually on the cell surface. They don't move the cell, they just move things across the cell surface. So if you look at that top image here, this could be in the respiratory tract actually moving mucus. Remember, mucus catches debris that you breathe in and is actually going to transport that so you can actually get rid of it. Flagella, obviously this is not found on every cell either, but this is a whip-like structure used for movement of the cell. Really, we'll just see this in sperm. And we're going to stop there because we don't need to actually review plant cells since we're actually studying the human body. I hope you're able to get all those notes, and tomorrow be prepared for an activity on this.